Hey, Mr. Williamson here, and this is the first of 5 videos on DNA and protein synthesis. This one's going to be about the introduction of DNA, a quick history on its discovery, and a little bit of information. Before we get started, make sure you're taking good notes. I personally like Cornell style. You can use outline, whatever you want, whatever makes your car go, use it. Here we go. Let's get started. You ever wonder why we all have the same body plan? By body plan, I mean, that is a human being. We all know that. We're not cockroaches. And yet at the same time, we all look different. Look at the people around you. Okay, if you're still at home, there's no people around you. But if you're in class, you'd look around and nobody looks exactly the same as you. You ever wonder why that is true? You didn't? Well, think about it. We do look the same. Nobody has a foot growing out of their butt. Okay, if you look on the screen, that guy in the third row, sometimes he does. All right, seriously, he does. But we all look the same. We all look different. Why? DNA. Deoxyribonucleic acid. And yes, you probably should get that down. That is the DNA stands for deoxyribonucleic acid. DNA, deoxyribonucleic acid. So what is this DNA? Where does it come from? What does it do? How does it work? And who discovered this evil way of making your life more difficult for the next 3 weeks? Well, in the early 50s, we knew there was a molecule that controlled the cell and all of its functions. We didn't know what it was, and they thought it was a protein. We knew about proteins, but we had no idea about DNA. Then along came 2 guys, Francis Crick, James Watson, working in England, Cambridge, England, and they were working on discovering the structure of an unknown molecule. They had some information. What information? Well, it had to do 4 things. All right? They knew it had to store a lot of information. All right? Think about this. Every one of your cells, and you have 37 trillion cells in your body. Every one of them contains the DNA to make one complete organism. That's a lot of information to store in DNA. Okay? It contains all the cell information on how to make every type of cell. Not just the body, how to make you a human, but how to make every type of cell. A liver cell, a kidney cell, a brain cell. Every one of them is stored in the DNA. All that information is stored in the DNA. Not only that, but it stores all the information on how to make every single protein the body needs. All of that stored in one cell's DNA. And you have 37 trillion cells. The human gene is about 5 billion base pairs. And that's not going to mean anything to you right now, but it will later. 5 billion base pairs long. It's about 6 feet, actually. All right? It contains enough information to fill 1,000 books. Your science book is only about 500 or 600 pages. We could fill 1,000 books, 600 pages, 500 words per page, and our books don't have that many words per page. That's like 300 million words. That's the type of information that DNA can store in one cell. The second criteria they knew. They knew that DNA had to be able to copy itself, had to be able to replicate itself with great precision. Okay? Think about this. Don't think about it too closely because there's a picture in there that you probably don't want to get in your head. But you started out as one fertilized egg. Sperm, egg, one fertilized cell. That was what you started at. And now we're 37 trillion cells. And in that one cell, though, was all the information on how to make all the rest of the 37 trillion. How did we get them into that information from one cell into 37 trillion cells? We had to copy it precisely every single time. We had to be able to copy it. So whatever they came up with in their idea of what DNA was, they had to be able to explain how it stored information, and had to be able to explain how it copied itself. They also had to be able to explain how it made mistakes, because DNA does make mistakes. Without DNA making mistakes, we can't explain, and I know, you're like, wait a second, you just said it has to be able to copy itself. Yes, I know. But without mistakes, We can't go on and explain evolution. We can't go on and explain cancer. We can't go on and explain genetic diseases. It copies itself. But somewhere along the line, we have to be able to explain, how does it make a mistake? And then finally, we knew it has to be able to be read. All right? We have to be able to get the information out of the molecule. Because a cell full of information that we can't access, we can't get that information out of, doesn't make any sense. It's like having a really nice car or truck or van or Razor scooter or whatever you got, but you're not allowed to take it out of the house. You're not allowed to take it out of the garage. You've got this really nice Lamborghini over here, and mom and dad, I mean, it's yours. No, it belongs to you. No, it's yours. Okay? And mom and dad won't let you take it out of the garage. And you're like, well, mom, but why? I want to show my friends. And no, it's safe in the garage. That makes no sense. Okay, well, actually it does, because you'd probably kill yourself driving into a wall at 150 miles an hour. Bottom line, though, you need to be able to access it. The DNA has to be, we have to be able to explain how it's read. Okay? We have to be able to read the DNA. They also knew a few things about it. It was really long, like I said, 6 feet long. They knew it was thin. They knew it consisted of 3 parts, and we'll go into these later: a sugar, a phosphate, and a nitrogenous base. And they knew that through X-ray diffraction. X-ray diffraction is a technique of where you crystallize a substance, and then you take an X-ray of it, and basically the crystals reflect the X-rays in a pattern to show you the shape of the molecules inside. Okay? They knew that. There's long, thin, large. They knew that it was made of a sugar, a phosphate, and nitrogenous base. But probably most important is they knew the nitrogenous base count of certain organisms. We can look at this table, and normally in class I would say, look at this table and see if you can see a pattern. I'm going to save us some time here. If you're looking, and you're looking at the box and seeing the pattern, the table and seeing a pattern, you'll notice that the innermost rows, these two, are all very similar in numbers. And these two are also very similar. So the outermost rows and the innermost rows are the same. Almost. Not quite, but almost. So they're looking at these different types of nitrogenous bases: adenine, guanine, cytosine, and thymine. Right? And they're looking and saying, well, 30.1 is real close to 30.4. And 28.7 is real close to 29. And 20.8 and 20.8, well, they're exactly the same. 21.8 and 21.8, exactly the same. They're very similar. And that's what Watson and Crick looked at. So they had, it has to be able to store information. It has to be able to replicate itself. It has to be able to make mistakes. It has to be able to be read. It's large, long, and thin. It's made of a ribose, a sugar, and a phosphate, and nitrogenous bases. And we know, they knew that the table that you're looking at here, they knew that table, the relationship between them. So what did they come up with? Okay. They also had one little bit of information, that the DNA was 2 nanometers thick. Now that doesn't mean anything to you, but it's 2 nanometers. It's consistent the whole way. And what they knew was the 2 purines next to each other. And you're like, well, what's a purine? Well, let me show you. This is a purine. The adenine and guanine are purines. It's a type of nitrogenous base. Cytosine and thymine are pyrimidines, another type of nitrogen. Don't worry about it. We'll get to that later. But they knew that. They knew 2 purines next to each other would be too big, and they knew 2 pyrimidines next to each other would be too small. What do I mean by that? Well, if you have 2 nanometers, all right, so this is 2. If you put 2 purines together, that's too big. If you put 2 pyrimidines together, oops, my bad, 2 pyrimidines together, that's too small. But if you put a purine and a pyrimidine together, it's just right. Looks like my uncle. Anyways, so it's just right. They know that it fits. All right? So they knew that the ratio of thymine to adenine was 1:1. We can go to the table and look at that. The thymine to adenine is 1:1. For every thymine, there's an adenine. And they know that for every cytosine to guanine is 1:1. Every one of these is 1:1. So what they came up with is double helix. The traditional, and you know what it looks like. Okay, the double helix. That's what they came up with. So let's review, and I'm going to go over the double helix in the next video. Let's review. They knew that DNA controlled the cell. They knew that it was not a protein. They knew it had a sugar, a phosphate, and a nitrogenous basis. There are 4 nitrogenous bases. They knew the guanine and cytosine was a 1:1 ratio. Adenine to thymine was a 1:1 ratio. They knew it had to be able to store information. They knew it had to be able to be read. They knew it had to be able to copy itself precisely every single time, and they knew it had to be able to be to make mistakes. They knew that the size of the molecules involved, the adenines and the guanines and the thymines and the cytosines, they knew it was 2 nanometers thick. And as a result, they came up with their double helix. Hopefully you took good notes. I will be glad to answer any questions you have. If you have questions, put them in the end questions on Edpuzzle or ask them in class. Go back and review anything that you don't understand. Like I said, this is just an introduction to DNA. The next video we're gonna look at, we'll talk about the structure. Hope you took good notes. Good luck.